Well, I'm excited to talk to you guys about integrated species distribution models today. So we'll dive in. So to give you an idea of where we'll be going today, I'll start with a kind of lightning introduction to species distribution models more generally, and the types of data that we use in species distribution models. Then I'll get into how we integrate these different data sets in these models, and touch on some of the appealing aspects of doing this data integration and some challenges and opportunities facing ISDMs. And then I've got several coding demos. I think I went a little overboard. There's 4 scripts on the GitHub repository. And so, we'll just, get as far as we can. And if we don't get through all those, they're there for you to go through on your own. And then we'll wrap up with sort of a Q&A time here. So to start off, I want to give sort of my general definition of a species distribution model, which is any statistical method that relates observations of species to environmental patterns. Really, the key part of this definition is that you have observations of species across multiple locations, and you're relating that to the environment in that location. When people hear about species distribution models, I think, what comes to mind are these sort of broad scale maps where, the abundance or occurrence of a species is predicted. So here we've got a great example with northern flicker abundance predicted across North America. This is from eBird data using, the eBird sort of very state-of-the-art species distribution modeling methodology. And that is certainly one common application of SDMs, is generating these, predictive maps across broad scales, continents, global. But that isn't the only application of SDMs. If you have data on northern flickers, for example, from one state, like, say, Iowa here, you can still use species distribution models to, relate your observations of these flickers to environmental patterns. So, often, this is a broad scale sort of exercise, but not always. And I also take the sort of broad view of species distribution models as a umbrella term that sort of comprises a whole bunch of different modeling tools. So, maybe you've heard of Maxent models, which are, I think that's maybe what people associate with SDMs most commonly, but really any method that, relates, spatially replicated observations of species to environmental patterns, I think fits within this category. So, methods like occupancy models and mixture models, even GLMs, I think can be used as, a species distribution model. And really the choice of the method that you, take, I think really is dependent on what type of data that you have available. And with species distribution modeling, there's kind of the big 3. There's presence-only data, presence-absence data, and count data. And so first of all, presence-only data is kind of what it sounds like. You have data on presences of species, but you don't have any information on absences. So the classic example is with museum specimens, where, for example, here we've got a nice tray of Lagellae bunting specimens. We have the specimens themselves, and we also have metadata, so we know where and when the individual was collected. But we don't have information on where, the collector went out and did not encounter a Lagellae bunting. So that's why this is presence-only data. And for I guess more contemporary example, a lot of community science programs might generate presence-only data. I'm thinking of iNaturalist, for example. And so presence-only data has the advantage of, you can often get a lot of it. There's large volumes of these data in some of these databases. But it has, potentially the disadvantage that In a lot of cases, these are opportunistic data. There may not be much of a sampling design in how they're collected. And also, we don't have that information on absences. So that brings us to sort of the next big category of data in the species distribution modeling world, which is presence-absence data. And here, again, we just have, information on presences, but we also have information on where a species is absent or where it is not detected. So a good example of a data stream that generates presence-absence data is camera traps, where, maybe you put out, 50 or 100 cameras across, some region. You may get detections of your focal species on, a subset of them, but, the cameras where you don't get images of your focal species are still giving you information. They're giving you that absence information. And so here we have the advantage of, having that information on absences. We don't have to, kind of guess at where the species is absent. And in a lot of cases, but not always, these presence-absence data sets may come from a bit more of a structured sampling design. So that's, potentially another advantage. So that was presence-only, presence-absence data. And now sort of the last of the big three is count data. And so here, we have those previous examples, presence-only and presence-absence. That was, binary. We didn't have information on abundance. And with count data, we have that sort of added dimension of abundance. So a good example here is, bird point counts, where maybe you send your field technicians out and they go out and survey, count all of the individuals within some radius of this point that they're surveying. So here again, we have information on absences because you can have, a count of 0. We have the advantage of having that abundance information. And again, oftentimes, but not always, you may have a bit more of a structured sampling design getting these counts as compared to, presence-only data. So those are sort of the three main data types that, we're often using for species distribution modeling. And inherent to all of those types of data is imperfect detection. We're just not going to be able to detect every individual every time we go out and survey. And so there's, it's recognized that this is, important to account for in our models, including our species distribution models. And so a couple potential methods to account for imperfect detection that I'll mention are, first of all, temporal replication. Here, for example, I've got a little example where you have multiple sites in your rows, and they're visited on multiple occasions, here with presence-absence surveys. And from those patterns of ones and zeros in your sort of detection history here, you're able to estimate detection probability and account for individuals that you may have missed when you were surveying. So that was one option. Another option that I'll mention is with distance sampling. So here, in addition to collecting, your count data or what have you, you measure or estimate distances to observed animals. And then you fit a detection function to those observed distances under the assumption that your ability to detect an animal declines over distance. And so in that way, you're able to account for individuals that were missed. So we'll see examples for both of these in those coding demos that I'll show you. So that was a lightning introduction to species distribution models and the types of data that are often used in species distribution modeling. And now I'll turn our focus to data integration and iSDMs. And so data integration broadly is the practice of combining multiple data sets within one model. And you may be familiar or have heard of integrated population models, or IPMs as they're often called. And so IPMs are an example of data integration. iSDMs are an example of data integration. So data integration is sort of this broad category. So all iSDMs are integrated models, but not all integrated models are iSDMs, I guess. And so before I go into specifics, I want to start with a metaphor here for data integration. So here I'm showing you a picture of, I don't know, back in the day, I think this is maybe like early 1900s. We've got a snowplow team here where we've got a pair of horses and a pair of oxen that are hitched up together to plow a road. And I think this is a lot like iSDMs, so or data integration. So here we're sort of combining forces of our horses and our oxen. And, maybe these, two animals have, sort of distinct advantages when it comes to snow plowing. Maybe horses have longer legs, they can get in deeper snow, and maybe oxen have, more endurance. So by, combining these animals into a team, you're able to get, more power and plow your road better. And same is sort of the case with data integration. Different data sets have sort of their inherent advantages and maybe disadvantages. And so by teaming them up, you can maybe expand or strengthen your inference. And I'll extend the metaphor a little bit further. I'm not an expert when it comes to horses and oxen and that sort of thing, but I assume that these animals require different harnesses. You can't just put a horse in an ox's harness or vice versa. And this is true with data integration as well. We want to acknowledge and account for the differences, among the data types that we're integrating. We don't want to just sort of indiscriminately throw them together. So just to formally give you sort of my definition of an iSDM, it's, a species distribution model that combines multiple presence-only, presence-absence, or count data sets, or, any combination of those in an effort to relate, spatially replicated observations of species to environmental patterns. And so it can be, I hinted at this a little bit with that metaphor there. It can pose a little bit of a challenge to kind of reconcile differences in data types when you're making an integrated model. So, for example, presence-only data and count data are quite different entities. In presence-only data, you have just the locations of individuals. Count data, you have, these counts, which are non-negative integers, corresponding to some surveyed quadrat. How do you make those work together in an iSDM? Well, sort of the way that you get these to work together is you assume that they are derived from or descended from a shared or common ecological state. So here I'm showing you, some landscape where we've got, the locations, the true locations of individual organisms. And we assume that, from this true state, we get our presence-only data as well as our count data. And then, we have built into our model sort of these distinct observation submodels. So we're accounting for differences in, how these data are collected and sort of the differences in format between the data. And so sort of how we find that common ground between these data types is, I think inspired in large part by, point patterns for iSDMs. So we assume that, all of these different data types that we might observe or might collect are ultimately derived from these spatial point patterns. So here I'm showing you a simple point pattern, the locations of individual organisms simulated across a landscape. And if we basically divide that landscape up into cells or pixels, the abundance of species within a pixel is just the sum of the number of points within, that pixel. And then from that, occurrence or occupancy, is just sort of a binary summary of this abundance data. So say you have presence only data and count data. You kind of think about, okay, what is the common ground here? They're coming from a point pattern, and so you kind of use that when building your model to make those data types work together. Now there's a realm of statistics related to point patterns and point process modeling, which I won't go into a ton of detail here because we often don't deal with these sort of spatial point process models directly. Those sort of are continuous space applications. They can be quite computationally expensive. And so, especially if we're doing sort of these broad scale modeling efforts, it really helps to make space discrete and sort of work in the realm of abundance or, occupancy. And as well, I think, these sorts of, quantities, abundance and occupancy are exactly what, we're interested in estimating or predicting with species distribution models. So I'll walk through an example of, how you make that integration process work in a model and sort of make different data types, kind of work together and sort of tell you information about sort of this shared ecological pattern. So with this example, let's assume that you have replicated count data as well as replicated presence absence data. So we've got those repeat visits for both of them. So first I'll start by talking about sort of the portion of the model where we might be modeling our count data. So this is going to be our submodel for our count data. First, we model the expected abundance, the lambda, across multiple sites, so indexed with I. So our expected abundance is this log linear regression where we have an intercept, beta 0, a slope, and then some predictor variable here. This could be climate, forest cover, whatever you want. And really, if you're doing a species distribution model and you want to make predictive maps, you'd probably want more predictor variables and maybe a spatial field here. But for simplicity, we'll just say we've got one predictor variable here. So that's sort of our model for our expected abundance. Then we model the true, latent abundance. We're not able to directly observe it across those sites indexed with I as a draw from a Poisson distribution, where the expected value is that lambda. And then our count data, which we have across our sites indexed with I, and then we've got repeat visits, which I'm indexing with J here. We model those counts with a binomial distribution, where the number of trials is the latent abundance, and then we have this detection probability here, which we estimate from those across those repeated visits, the patterns in those counts. So this is really just an N mixture model, and this is sort of what we're using as our submodel for our count data. So next, I'll walk through sort of the submodel for our presence absence data. And here I'm going to index our sites where we have presence absence data with T. So this is to emphasize that these presence absence data may come from different sites or a different number of sites as our count data. So again, we start out by modeling the expected abundance of our sites indexed here with T as just this log linear regression. And this is the really key point here is we have these same parameters, this beta 0 and beta 1. So our intercept and our coefficient for this predictor variable X. So I'll just flip back here. So we've got beta 0, beta 1 here, and we've got beta 0 and beta 1 here. So really this is how we accomplish this data integration through, this is called a joint likelihood. Really the name of the game is, same parameters showing up in multiple places within your model. So now we have to do sort of a currency exchange to go from the land of abundance to the land of presence absence data and occurrence or occupancy. So we model our the latent occupancy or occurrence state of those sites with a Bernoulli trial, where the probability is this function of our expected abundance lambda. So 1 minus e raised to the negative lambda. This is sort of what lets us go from living in the land of counts and abundance to the land of presence absence data. And then last of all, we model our our presence absence data, y here. So y across our t sites and j repeat visits. We model that with another Bernoulli trial where the probability is detection probability times the sort of latent occurrence state here. And so really the things to note here is we have a shared model for the latent ecological state or pattern. So we have this model for sort of the expected abundance, which is shared between those two submodels. And then in this case, we are assuming that we have separate observation models. So we've got this detection probability PD for our presence absence data. And in our count submodel, we're estimating this detection probability P sub C for our count data. So same ecological state, but different observation processes in this example. So with that simple example, I'll sort of transition to a few points of why you might want to use iSDMs. The first example I'll give is that I think these iSDMs give you a lot of latitude to be creative with what constitutes species distribution data. So this is an example from a recent paper that I really liked, where the authors combined presence absence data from camera trapping, which is commonly used in species distribution models. They combined that with these presence only wildlife complaint data. So they were working with City of Chicago. They basically got information from the city on whenever folks called animal control to complain about raccoons or opossums, that sort of thing. And they considered those presence only data. They put that in an iSDM with their camera trap data, and with that they're able to make these really cool maps predicting spatiotemporal patterns of human wildlife conflict. And so really that's sort of a exercise that you're really able to do with this iSDM framework. So I think that's a really cool example. Another appeal of iSDMs is, in a lot of cases, you may be able to increase the accuracy and precision of, your estimated quantities of interest. So this is, just a simple example, simulated example, where we have a landscape with presence-only data, as well as, some distance sampling data, which allows us to get at, to estimate detection probability and account for imperfect detection. So over here, you'll see that, when you don't have any distance sampling included in a model, when you're only using presence-only data, your estimate of abundance here is biased. So this blue line here is the true value. But adding in even just a small amount of distance sampling data allows you to estimate that abundance accurately. And as well, the more distance sampling data that are included, the greater precision you're able to get at estimating. I believe this is a covariate effect here. So really with this data integration, we're increasing our sample size essentially by putting multiple data sets together. So that's another big appeal of iSDMs. Another appeal is this idea of expanding your inference. This could be expansion of your inference across space, time. And here I'll just give you a hypothetical example of, maybe you have presence-only data that stretch back for several decades. And then you have replicated count data, which are, maybe higher information content, more structured data. You have these replicated counts, but for a shorter time span. Well, theoretically, you could, specify an integrated model that combines these data sets, and you, in essence, sort of inoculate your sort of longer term data with some detection information from your shorter term count data. And with that, you're maybe able to expand your inference and estimate quantities like abundance for this, whole sort of time series. So those were some of the appeals of iSDMs. And now I'll just touch on some of the challenges and opportunities facing sort of implementation of these models. The first challenge is this assumption that these data sets are telling us something about a shared ecological process. So in the example I walked through, for example, we had those shared parameters in our submodels for those two different data sets. And I think it's really important to evaluate that assumption because if you're comparing data sets that are collected maybe from very far apart places or far apart in time, you may be in essence comparing apples and oranges if you're assuming that quantities like species environment relationships are the same across those data sets. So this is an important assumption to evaluate, and I think when you're designing a study and thinking about applying an iSDM, this is really the first thing you have to think about. Another challenge, I guess, is sort of this tension between increasing the volume of your data and making sure you don't perhaps overwhelm a structured data source you have with large volumes of low information content or biased data. So this is a great study where these authors performed a bunch of simulations and showed that if you combine a bunch of biased presence-only data with your structured count or presence-absence data, even with an iSDM, you're still not going to be doing too well because that bias in your presence-only data might just kind of really throw things off. So that's another thing to really evaluate when you're designing a study. Another big challenge is model selection and model validation with iSDMs. And I've left this slide blank here to kind of illustrate that it's really uncharted territory. If you want to see how much information you gain by integrating a data set into your model, you can't really compare that integrated model with a non-integrated model using AIC or other sorts of information criteria, because those sorts of tools assume that your models that you're comparing have the same response variable. Same thing with model validation. How do you think of what model fit is when you have multiple data sets? Do you compute model fit relative to one data set or the other or both? So I think there's a lot of opportunities for growth here. And then the last challenge, and really I think this one is maybe more of an opportunity, is that these integrated models and integrated species distribution models, in a lot of cases will require you to, learn to code in languages like Nimble or JAGS or Stan or Bugs, these sort of custom coded Bayesian modeling languages. That's certainly a bit of a challenge and maybe a barrier to entry, but I think it's a bit of an opportunity. But that I think is changing a little bit. There are some R packages showing up on the scene that are allowing users to fit integrated models with sort of relatively user-friendly interfaces. So for example, Jeff Dozer's SP occupancy package has functionality to fit integrated occupancy models, the models fitting or integrating multiple presence-absence data sets. And he gave one of these webinars a few months ago on that package, so check that out if that's interesting to you. And another example that I just learned about is this pointed SDMs package. which looks like there's a lot of great functionality to fit integrated species distribution models in a pretty user-friendly interface. So I haven't had a chance to look into that one yet, but that looks like a really promising tool as well. And so on the topic of coding, the coding demos that I'll be showing you here in just a moment are in the Nimble language. And so if you want to follow along and run the code that I'll be using, you'll have to have the Nimble R package installed. And as well, you'll also have to have a C++ compiler set up on your machine. So if you need to do that, the installation instructions are in the Nimble manual, which I've got a link to here. So if you need a compiler, go ahead and get that set up if you want to follow along. And so I've got 4 coding demos set up today. I think we'll probably really only have time to get through the first, maybe 2 of them. And so the first demo that I'll be sharing with you is an iSDM that combines replicated counts with single visit presence absence data. And so this is an example where, maybe you have, sort of count surveys where you're going out and surveying a pretty small number of locations within your focal landscape. It's quite a bit of effort to go and do these repeated surveys, so you're not able to, cover a lot of ground spatially. So maybe then you complement that with, these single visit, presence absence surveys where you go out once and do, a quick sort of rapid assessment. So that's our first example we'll be looking at. The second example is an iSDM that combines presence only data with replicated counts. The third example, which I don't think we'll have time to go through, but the script is there and the GitHub repository, is a model that combines single visit counts with distance sampling data. And then the last example is a model that combines fine resolution replicated presence absence data with coarse resolution single visit counts. So this is an example that I kind of made based off of some work that I did where I integrated camera trap data, which were collected from points, with harvest records, which are sort of summarized at the county level, so a much different resolution. And this model accounts for that mismatch in spatial resolution between data types. So with that, I will move over to my R script. So I'll be starting here with this example 1. And so this is an iSDM that combines replicated counts and those single visit presence absence data. And so I think, maybe the easiest way to do this is I'm just going to run the code here to simulate the data, and then we'll look at some visualizations of the data so you have an idea of what the data are going into this. But I won't sort of go through in excruciating detail of how I'm simulating these data. So if you're following along, I'll be running lines 1 through 122 here. So we'll get that running. Should hopefully only take a moment or 2. Looks like I had some extra stuff in there, so let me run that again. Okay, so we will look at the visualizations of these data. So I'll go back to this first plot. Let me make this larger. So what I've done here is I've simulated a point pattern across a hypothetical landscape here. So you see all of those points across this landscape. Those are representing the locations of hypothetical individuals. So for species that don't move around. So, plants, for example, that's the location of the plant. For, animals that move around, this could be, the snapshot locations of those individuals at a moment in time, or it could be, the average, activity center over time. And I've simulated this so that the intensity of this point pattern increases from left to right. So there's sort of more points over here on this right side of this landscape. And then I've also sort of overlaid this with a discrete landscape where I've got these grid cells laid across the landscape as well. So next, I'll show you our count data. So here, a few things to notice are that we have repeated visits. So we got 4 repeated visits. Another thing to notice is that we have a relatively small number of those cells within the landscape that we survey with these counts. I think it was like 3 or 5% or so. And then another thing to notice is that we have imperfect detection here. So, I'm showing you the locations of these individuals sort of in the background, but we're not able to observe those directly or perfectly. So for example, I'm looking at this point here in sort of the top middle. There is an individual in that cell, but in this third visit, it has not been detected, but here it is detected on that 4th visit. So we have imperfect detection simulated in these data. And then sort of the last plot here is showing you the presence absence data. So here we have we've got a single visit. These are all different cells from the count data that are surveyed. And you'll see that there are relatively more cells or quadrats that are surveyed. We have only presence absence data. So, we have cells where there are multiple individuals, but still only gets a 1. For the species being detected there. And here as well, we have imperfect detection. So I'm looking at, let's see, some of these cells on the far right side where there are individuals in there, but they are not detected. So that is sort of the data that we're working with. And so now I'll sort of get into the model and how we sort of model those together. So the first thing that we have to do when we're using Nimble is we have to package up our data and what are called constants to be used in the model. So I will run this and then we will look at those. So first, this data list. This is a list of 3 sort of data objects we'll be using in our model. We've got this detection object, which is a vector of our presence absence data. It's just a vector of 1s and 0s. We have our count data, which is a matrix. So we've got 29 sites where we have count data and those 4 repeated visits. And then we have this x-coord object, which is just the x-coordinate of each quadrat's centroid, which we're going to use as a predictor variable in our model. So that was our data. And then the constants list just gives us some information that will be used for indexing in the model. So det ID and count ID are the cell IDs of cells where we have the presence absence data and the count data. We've got the total number of pixels and pixels here. We've got the number of pixels with count data, that's 29. The number of repeat visits, which is 4. And then the number of pixels with our presence absence or detection on detection data, which is 208. So that's the setup. And now I'm going to go ahead and just run this model so it can be running while I'm talking about it, because I think it might take a minute or two. So I'll just run all of that. And then I'm going to scroll up and talk about the model itself. So here, starting in line 124, we have the code for the model itself. And once we get inside of this bracket here, we are no longer in the R programming language. We are in the Nimble language. And so, one thing to note that, I found confusing at first when I started using these sorts of programs is that this is a declarative programming language. So it's not like R, where the order of operation matters. It really, you can put whatever lines wherever within the model. It doesn't matter what order you put in. So this is our Nimble model, and I'll start in this section here. We have a model for the shared ecological state across this landscape. So what we're doing here is we are looping through all of the pixels within that landscape. We have this model for the expected abundance, this log linear regression, very similar to what I showed you before. We've got an intercept, a slope. We have the x-coordinate here as our predictor variable. And then we model the latent abundance within that cell as a draw from a Poisson distribution with the expected value set to that lambda. So that is sort of our model for the landscape there. And then we have a submodel here for our replicated count data. We loop through the sites where we have counts. We loop through the repeated visits, and we model each count with a binomial distribution where we have this detection probability P here, which we're estimating. And then the number of trials is the latent abundance within that cell. So that is our submodel for the replicated counts. And then we have this submodel here for the presence absence data. So we loop through the cells where we have the presence absence data, and we model those data as a Bernoulli trial. And here we're assuming that we have a shared observation process. So we're using the same detection probability as up here. So in essence, these count data are sort of doing the hard work. They're allowing us to get this estimate of detection probability. And then that is borrowed down here. And so we multiply that detection probability by that expression that I showed you earlier, this 1 minus e raised to the negative lambda. So the important thing to note here is that both of these data sets are telling us something about or giving us information on the expected abundance patterns within the landscape. We have latent abundance showing up here in our count submodel, and we have the lambda showing up here in our model for the presence absence data. So then last of all, we could estimate the total abundance within this landscape by summing up those abundances. And then I didn't mention it, but we've got priors up here for our parameters, and I'm using some sort of my default kind of weakly informative priors there. Okay, so that was sort of the code, and it looks like it finished running here. I'll just note here that with using Nimble and these other sorts of Bayesian modeling languages, you have to provide initial values for your MCMC algorithm. So that's what I'm doing here. You have to tell the model what parameters you want it to track. And then since this is run, I will just look at this output. So here, what's printed out in the console is just a summary of the posterior distributions of the parameters we wanted to track. So detection probability here, P, I believe I used simulated the data with P set to 0.5. So it looks like we're estimating that pretty well. It looks like the 95% credible interval here overlaps that true value. And then the total abundance, let me just remind myself what that was. So there are 404 individuals simulated across that landscape, and we get this estimate of 362, but again, that credible interval overlaps that true value. And then these last two columns here, this R hat and N dot F, are convergence diagnostics. So typically we would want this R hat to be about 1, and we would want this we would want a pretty high number for this NF parameter here. So that's telling me that this model has converged more or less. Okay, so that was the first example, and I think based on timing here, I think I've got time to run through the second example here. So go ahead, and if you're following along, we'll switch over to example 2. I'm going to go ahead and clear out my R environment, and I'm also going to restart R just so we have a clean slate to start with. And so the second example is an iSDM that combines presence-only data with replicated counts. And this model I've adapted from the Applied Hierarchical Modeling in Ecology, second volume. So really all I've done here is I've taken their JAGS code and I've converted it to Nimble. And so we're going to use a data simulation function that they provide in the R package that accompanies the book. So a big shout out to Andy Royle and Marc Kéry for that awesome book and for this example here. So what I'll do here is I'm just going to run lines 1 through 23 here. This function here is going to simulate the data we'll be using. And I'll do a similar thing where we look at the visualizations of the data, and then we'll get into the code itself. So first of all, we have on the left here, I'm showing you the point pattern that is simulated. So all of those little points across this landscape are locations of individuals. And on the right here, I'm showing you the presence-only data. So those points are individuals that are in our presence-only data set. And sort of a key aspect of this model is that it assumes that we have at least one unique covariate that describes variation in abundance. So here I've got that covariate shown in the background here, as well as at least one unique covariate that tells us something about sampling bias or detection bias. So you'll notice with this presence-only data, it seems like there's more presence-only records over here on the right side of the landscape, even though that there are a lot of individuals over here on the left side. And so practically speaking, if you're working with something like iNaturalist data, this sort of bias or detection covariate might be something like human population density. And then these two covariates here that I'm showing you in the background are, these are at a fine spatial resolution that we're using to approximate continuous space. All right, so we'll look at the next plot here. So here we have our count data, which is collected at a coarser resolution where we have these quadrats across this landscape. So here on the bottom left, I'm showing you the true abundance within each cell. So that's just the sum of how many points there are within each one of those cells. And then the bottom right we've got a summary of our count data. So you'll notice that we have a pretty small number of quadrats across this landscape that are surveyed. And then last of all, we've got those same two covariates I mentioned previously, but aggregated to this coarser scale. All right, so now we will package up our data and constants like we did before. And we'll take a look here to see what we've got. So first, our data object, I'll just walk through here. This Y object is our presence-only data. So this is for that fine resolution landscape that was shown in the previous plot. So we have a 1 for each cell wherever we have a presence-only record, and then we basically fill in these pseudo absences, these zeros, for cells where we don't have a presence only record. Log area is the log of the area of those fine resolution cells. And then we have those two covariates for that finer resolution grid. So our covariate for abundance and our covariate for detection or sampling bias. Then we've got the replicated counts. So we've got 25 sites that are surveyed, 3 repeat visits. And then we've got those same covariates I mentioned previously, but aggregated to that coarser scale for those cells where we have the count data. And then area is the area of those coarser resolution quadrats. And then the n quadrats is the number of those coarser resolution cells across this landscape. So now I'll just show you the constants. What we have here is n pix, that's the number of pixels in that fine resolution landscape. N site is the number of sites where we have those replicated counts, so that's 25. And then n surveys is our number of repeated visits for the count data. Okay, I'll do the same thing as before where I get the code running and then I'll walk through the model a little bit. We'll get that going. And so here we have our model again in Nimble. I'll start out here. We have a model for our fine resolution grid cells. So we loop through all, I think there's like 10,000 of those cells. We model our presence only data with those pseudo absences as a Bernoulli trial with probability psi. And then here we model that psi as a function of the area of the pixel, log of the expected abundance, and then log of this P1, which you can think of as sort of a thinning probability. How likely is an individual to be thinned and appear in our presence only data set. And so important reminder for these parameters to be identifiable, the lambda and this P1, we have to have at least one unique covariate for each of those. So here we've got our model for that lambda, the expected abundance. We model that as a function of an intercept coefficient and then that X covariate, or our covariate, which describes variation in abundance. And then we have this model for the thinning probability. And we model that as a function of, again, an intercept, this coefficient, and then that bias covariate. So again, for practical applications, that bias covariate may be something like human population density or road density, some sort of variable that you think is associated with how observers are going out and collecting your presence only data. So this is sort of the submodel for your presence only data. And here we have a submodel for our replicated counts. So we say that the latent abundance within each of those sites that we survey with counts, we treat that as a draw from a Poisson distribution, where the expected value is the area of the quadrat multiplied by this lamb parameter, which is the expected abundance. We model that lamb with this log linear regression, where we have the exact same parameters showing up down here. So beta 0 and beta 1. And then we have this covar X, which is the same as X covar, or this X cov. It's that covariate that's associated with variation in abundance, but this is aggregated to that coarser scale there. So very important here, we have those shared parameters. That's what makes this an integrated model. And then last of all, we loop through our repeated surveys. We model our counts with the binomial distribution. where the number of trials is that latent abundance. And then we have this detection probability P2 here. And we have a model for that as well. And so here, sort of the key thing to take away is, we're making inference on this shared ecological pattern, this sort of shared pattern of abundance, or really the intensity of that, point pattern across that landscape. And we have separate observation processes here. So we're estimating this thinning probability here, which is different from this detection probability here. Okay, and then last of all, you can calculate with this model abundance for the landscape one of two ways. We can either sum up the expected abundances within those fine resolution pixels and multiply it by the area of those fine resolution pixels. Or we could just, essentially get the average abundance of those surveyed cells here and multiply it by the number of cells that we survey. Okay, so it looks like our model ran. So I'm just going to jump down and look at the summary of the posterior here. So here I've only run one MCMC chain, just to make it go quickly. Typically you'd want to run multiple chains, so that's why we have these NA values for R hat and N.F. And if we go back to the previous plot here, you can see in reality there are 1,842 individuals, and it looks like our estimates, sort of using those two different methods, are giving us estimates that overlap the true value. And then if you scroll up farther to where we're simulating the data, it looks like all of these parameters are estimated quite well. We've got -2 for alpha 0 is the true value, and our estimate is -1.9, and so on and so forth. All right, so that was our model that combines presence-only data with replicated counts. Looking at the clock, I think I'll sort of not go into the other examples for the sake of saving time for our Q&A. I'll just briefly mention this third example is a model where we have counts as well as distance sampling data. So if you're interested in so that sort of application of using distance sampling, this would be a good one to check out. And then this last example is a model where we have fine resolution presence absence data and then coarse resolution count data. And again, this is a script I wrote that was sort of modeled after that example I showed you about the camera trap and harvest data. So if you're looking at this code and you see, talking about harvest and cameras, that's why that's there. So I think with that, I will hand it over to Dennis to sort of moderate the questions, and I will happily take questions that folks have.
All right, thank you very much, Neil. Great presentation. So I'm looking here at Poll Everywhere again. If people have questions, please include it in that website. I'm just putting it here in the chat for everyone. And you can also go and upvote the questions that you want to hear the answers for. So Neil, the first question is, can you offer some insight regarding kind of what are the considerations, what are the things to consider when thinking about determining the spatial resolution of these integrated species distribution models?
Yeah, that's a really good question, because fact of the matter is that in the real world, a lot of these data sets that we may have that we want to integrate sort of exist at different resolutions. So how do we reconcile that? I guess my sort of initial sort of response to that question is thinking about, I would think about thinking about what we have, what data set do we have at sort of the finest resolution, and kind of build up from there. So for example, if you have, surveys that, you're surveying, pretty small grid cells, maybe half a kilometer by half a kilometer, sort of specifying that as sort of your base in your species distribution model, and then sort of building up from there. But I don't think there's an easy way to go about that. But my first sort of reaction is start with sort of your finest resolution and go from there.
Mm-hmm. Great. And then there's a question here of, in terms of how to quantify uncertainty in iSDM.
Yeah, so good question. I'm not entirely sure what sort of uncertainty the question asker is asking about, but a few thoughts. I think the first example that comes to mind is this idea of kind of bias and how much you kind of want to how much emphasis you want to place on different data sets. So there you may have a data set that's really structured, you have good spatial sampling, you have some sort of information that allows you to estimate imperfect detection. If you have a data set like that and you want to combine it with one of these larger sample size, but maybe opportunistic data sets, can you tell your model to give more weight to the structured data set. So that is something that is possible. I think I have seen some papers that kind of weight the data sets based on how much you want them to affect your inference. So that's sort of one sort of application of uncertainty, because you may have you may have different levels of trust or sort of confidence in your various data sets. I guess in terms of uncertainty in parameters, with these Bayesian models, you're getting whole posterior distributions for the parameters you're estimating, rather than sort of point estimates. So for example, in this this summary I have here still up on the screen, you're really getting a full distribution for all of these parameters that are in your model. It's not just one value. So in that regard, you do have uncertainty, and that's sort of carried forward into any sort of parameter that you would estimate. So abundance or occupancy, that sort of thing. But definitely a very relevant question, and one that's important to think about is this idea of uncertainty. And maybe if the question asker was thinking of something else, they can sort of chip in and sort of clarify what they were thinking there.
Mm-hmm. Cool, great. And then I think this is a question that, and it's the one that has the highest number of votes here. That's an important question is, can you make any recommendation regarding courses, papers for someone who has never completed species distribution models except, say, generalized linear mixed models, GLMM, but wants to learn more about species distribution models? What sort of resources would you have to suggest for people that want to learn more?
Sure. So I think the first thing that comes to mind are those books I mentioned, the Applied Hierarchical Modeling in Ecology. Actually, I've got them right here in my office. So there's 2 volumes, these books with dragonflies on them. I think these are a really great resource for ecologists who want to get more into quantitative ecology. They cover everything from capture recapture to species distribution models, occupancy model type things. So you can sort of pick and choose what chapters you want to focus on. So, it's these big, thick books, but, there may be just 1 or 2 chapters that, are especially relevant to what you want to do. And then as well, they have, each volume has a couple sort of good general chapters sort of talking about, statistical inference in general and data simulation. So I think that would be my number 1 recommendation for, a place to start. And then in terms of, papers you might want to check out, a few recent ones that I really liked are, there's one in Trends in Ecology and Evolution by Nick Isaac and others. I can't remember the title, but it was, something about data integration and species distribution models. And then there are several other recent, like, helpful, accessible reviews about iSDMs. There's one by Rob Fletcher in, I think it was Ecology. There's one by David Miller in Methods in Ecology and Evolution. And those are all like within the last, 4 or so years. But yeah, I would start with those books I mentioned and then work out to sort of papers from there.
Great. So this question starts with a compliment. Great job. Awesome. So an overriding concern with presence-only modeling is correctly accounting for sampling bias. Can you please comment on the importance of doing that within iSDMs when using presence-only data? Do iSDMs provide any spatial capabilities in dealing with the problem of sampling bias?
Yeah, that's a really good question. So, I guess my first thought on sort of this question is, what are you getting out of the model? So, I think if you have, data that are maybe biased and you're not really sure, how, they're observed, how they're sort of, derived from that, latent state. I think you have to sort of think about and ask yourself, am I really going to be able to estimate quantities like abundance with these data, or can I be, satisfied with some measure of like relative abundance or relative probability of occurrence? So that's sort of the first thing that I would think of is, I think, some types of data and some of these models, may not really be able to give you, those sort of, measures. You may have to be satisfied with these sort of relative measures. I think, yeah, in terms of like presence only and bias, like I think that's a super tricky thing and something that I think, I honestly don't know a ton about, but my general thought is, if you put a lot of bad data in your model, that's probably going to affect, what inferences you're able to make. iSDMs are, it's not a magic bullet. There's a lot of cool things you can do with them and a lot of appealing aspects, but at the end of the day, if you put in a lot of really biased presence only data into them, I think you're not going to have, unbiased results. So I don't know. And I guess I'm just looking at the question, can ecological niche models combined with iSDM estimate? Okay, yeah, I think I'll move on to the next question. But that's a good question, and yeah, I'll have to do more pondering about it.
Yeah, it seems like one of the challenges, at least based on the example that you gave, would be like if you miss an important covariate, right, that influences the detection bias, then what happens? Are you better off just using the data that you're most confident about, or bringing this other data set with the risk of potentially not accounting for the sampling bias appropriately, right, and ruining everything? I think that's probably the question.
Yeah, and I think that's a sort of with that last example I showed you guys, like that's sort of a very strong assumption, is that you have that sort of information. And I think in some cases you can kind of make or make a case for, oh, this information we have is associated with spatial variation bias, but in practice, that can be a tough thing to do. So yeah.
Mm-hmm. Yeah. So the next question that you kind of read is, can ecological niche models be combined with iSDM to estimate species or community abundance and reduce errors from presence-only data set?
Yeah, so that's a good question. I guess my thought is I typically think of niche models and ecological niche models as being another name for species distribution models and SDMs. In a lot of cases, I think that these environmental niche models are kind of more in the presence only sort of sphere of species distribution modeling. So I guess I would say that I guess I would say that environmental niche models are kind of maybe a subset and sort of specialty of these species distribution models, but I don't think that there's necessarily a way to I guess I wouldn't consider them like a distinct sort of class. So I don't really know what to say about that, but
that was similar to what I was thinking as well. What about, do you have any thoughts to share regarding spatially filtering your data when you know that it has been sampled in a biased way?
Yeah, so again, this is sort of in the realm of sort of the presence only modeling practices. I know that there's some algorithms where you'll have some sort of thinning operation that you do prior to putting your data into the model. And really sort of with this example that I've shown you, this sort of second example, you're kind of doing that, but within the model by having this covariate that's sort of describing, variation in spatial bias. So, sort of doing that prior to modeling is, I think would kind of be a bit of a similar process, right? You have some idea of, how this bias is arising, and you maybe try to mitigate it by, I've seen examples where people sort of calculate some sort of, no points within, 10 kilometers of each other are allowed. And, you sort of have some sort of thinning algorithm. So, I'm not sure like if that approach versus sort of the in-model covariate approach, which is better, but, I think I've seen, mostly sort of these distance-based sort of approaches where you set a rule of like, oh, no point within a certain, radius of another one. But that's a good question.
And well, this is a tricky one. Are you able to briefly talk about model transferability in iSDM? So if you are going to use that model to a new environment, a new area, or a different period of time, what is your perspective regarding model transferability?
Yeah, that's another really good question. I think, I guess proceed with extreme caution, I guess would be my, advice, because, with these sorts of models, you're assuming that some of these quantities that you're saying are shared or informed by both data sets. You're saying that they're the same between, the places where you have data or the times where you have data. And so even more so, if you're interested in using these models to project to the future, you're making the assumption that quantities like, the association between a species, species abundance and some environmental predictor, you're assuming that's going to be exactly the same and never change, if you apply it to a new spot or, forward in time. So I guess I don't have like a ton of practical advice in terms of, how to, do the, sort of transferability and, how to evaluate that. I guess I would just, really caution, be aware that you're making this assumption of sort of shared abundance patterns between, areas where you're applying the model. And, you may be able to account for that in some ways. Maybe you have, for example, if you're interested in transferring in time, maybe you have something like a random effective year that you incorporate into the model. But yeah, that's really tricky waters getting into that. So yeah, good question.
Yeah, it seems like this question is really also associated to that slide when you were talking about extending inference, right? You had one data set that spanned multiple decades and another data set that was just a given decade that was a more precise data set. But then if you were to then make inference for the entire multiple decade period, then it seems like we could run into issues like that are related to this question, right? Of model
Absolutely. Yeah. And so like, just for, one example, I've been working with, a data set where we have this like sort of long-term, I think like 20 or 30 years worth of, single visit counts. So we have these count data, but we have no sort of temporal replication. We don't have any like distance sampling information. So there's no way to evaluate, imperfect detection. And then we have basically 2 years of distance sampling from the same system, collected in a similar way. So we have counts plus, the distance measurements. And so, thinking about like, can we, take this detection probability that we're estimating from these 2 years and apply it to, 30 years worth of data. Just in conversations with the folks that, collected the data, it seems like, habitat was changing, sort of some of the settings were, changing in terms of, land cover, that sort of thing. And so that was probably influencing, detection probability. And so, it's just really, I think you have to have pretty strong justifications to, do some of these sort of transferability practices with these models, but really cool to be able to do it though.
Yeah. All right. So there are a couple of additional questions that unfortunately I don't think we're going to have time to go over because now it's already 1:30. But I think what they overall show that there are a lot of very interesting questions and a lot of interest in the community regarding integrating these different data sets, how to deal with bias, one data set being much bigger than another, how to weight them. So this field is really very exciting, and I think this presentation was very helpful. So I would like to thank you, Neil, for making this presentation and creating these 4 examples. I think the code is going to be useful for people that want to follow up on this and learn more. And so I really appreciate your effort in putting them together.